Hello, everyone, and welcome to today's webinar. I'm Melissa Romero, Forensic Science Consultant in CNN Media Group. I will be moderating today's event. This webinar is titled Enabling Drug Discovery with Diverse Lead Generation Platforms. This event is sponsored by WuXi AppTec. CNN works with sponsors to identify topics of interest and value to CNEN's audience and consistent with CNEN's mission to provide news and analysis of the chemistry enterprise in a timely, accurate, and balanced fashion. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please look at the Help widget at the bottom of your screen or type your query into the Q&A box. If you are disconnected during the webcast, please log in again according to the instructions you received earlier. You're encouraged to contribute to the success of this webinar by asking questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will be posing as many as time permits. This webcast allows certification of participation. To receive your certificate, please click on the certificate icon below your Q&A box to download your copy after watching the webinar. Please note that CNN does not endorse any company, products, or services that may be mentioned in the webinars. Each webinar will be archived at CNN Online after the live webcast. The presentation today is sponsored by WuXi AppTec, a company that provides a broad portfolio of R&D and manufacturing services that enable organizations in the pharmaceutical, biotech, and medical device industries worldwide to advance discoveries and deliver groundbreaking treatments to patients. With industry-leading capabilities such as R&D and manufacturing for small molecule drugs, cell and gene therapies, and testing for medical devices, WuXi AppTec's open access platform is enabling more than 4,400 collaborators from over 30 countries to improve the health of those in need and to realize the vision that every drug can be made and every disease can be treated. During the presentation today, we will be hearing from Dr. Nushka Shamar, Dr. Penny Jha, Dr. Saskia Vininger, and Dr. Ling Zhu. And now let's begin with Nushka.
Thank you, Melanie, for this kind introduction. In this webinar, we will briefly discuss evolution of lead generation toolbox in drug discovery, which enables fast innovation nowadays, key tools and strategies that enable us to work in this space, and the state-of-the-art platforms for hit-to-lead generation will be introduced by leading experts at WuXi AppTec. Penny will briefly discuss ACMS screening, Saskia fragment screening, Ling will talk about virtual screening, and I will introduce DNA encoded library screenings. Prior to 2000, the most frequently employed lead generation strategies were random screening and rational design. In those days, high throughput screening was seen as the major breakthrough and actually enabled about 10,000 compounds to be screened per day. And now imagine, within 20 years, we were able to come from 10,000 compounds per day to over 80 billion compounds per day. So how is that possible? Next to rational design and classical HDS, which still exists today, new methodologies became much stronger, and disruptive technology like DNA encoded libraries enabled the screening of these 80 billion compounds in a day. State-of-the-art lead generation toolbox is composed of 5 main methods. HDS screen, as I mentioned, is historically very important, but we will not discuss it today. We will focus on ACMS screen, DEL screening, fragment screen, and virtual high throughput screen. Different target types require different hit finding technologies to find the best hits, and different methods will uncover different chemotypes. So we will discuss pros and cons of focusing only on one technology, or in the advantages and synergistic effects when we combine several technologies in one bundle. Here's a brief comparison of screening methods. Waste chemical space can be introduced and investigated using these methods. HDS usually has 10 on 6 to 10 on 7 library size. ACMS can go up to 1,000,000 10 on 6. Fragment screen is actually the screening method which uses practically very tiny libraries composed only of 1,000 or maybe 10,000 small fragments. Virtual screen can screen compounds in the number over 1 billion. And of course, the DNA encoded library are actually exceeding all the imagination in the numbers of compounds screened. We can actually screen several billions of compounds. The targets can be very different, can be proteins, can be nucleic acid. Depending on the method used, we can either screen for binding, as in the case of ACMS, TEL, or virtual screening, or also based on the functional activity, like in HDS or even fragment screen. Some of those methods, of course, they come with some drawback because we have to immobilize the target. In some cases, we don't have to immobilize it. And overall, the summary of pros and cons actually then decides or determines which targets are preferably screened by which method. Nowadays is, of course, everyone understands that practically you need really highly sophisticated composition of any library if you want to get reliable and good hits or novel hits innovation to find novel chemical matter. Sometimes actually the protein quality diminishes out of, let's say, of the focus. But to this point, I would like to remind you, as Frances Arnold said, you get what you screen for. So no matter what your screening method is, which you will use to tackle your challenging target, please focus on the target quality. It should be highly pure. It should not aggregate. It should be as monodispersed as possible, even if it has some post-translational modifications, and it has to be correctly folded. So very often, of course, we are dealing with novel targets for where no ligand is known. So we do not have positive control in the form of small molecule. Still, we can check the integrity of the protein by binding, for example, to his interaction partner. Preparing protein for screening purposes is a multi-layer process. At the very beginning, you should design your target protein with the screening goal in mind. And with the clever design of your target protein, you can satisfy the needs of several different methods using practically the same protein construct, which is of huge advantage when you want to compare the data, the results across different methods and validation technique. So you have to ask yourself, if your protein needs a cofactor, what kind of specific tag you need, if you need to label it with a dye, what kind of isoform you want to have, if you want to investigate mutant, truncated form, complex, if should be practically dephosphorylated, deglycosylated. As you see, this is a very complex story. And in the case that you cannot really decide what would be the best way, we highly recommend starting with several constructs in parallel, investigate them in parallel, and this can actually reduce an overall project timeline for several months. So, Affinity Selection Mass Spectrometry is first of methods which will be introduced today. So, dear Penny, this ACMS technology is a novel technique enabling the discovery mostly of small molecule ligands for challenging targets. Can you please briefly comment also on the protein requirements before we dive deeper into the matter?
Thank you for the question. Yes, the protein quality will indeed affect ACE mass screening, as I'm going to talk about in the next few slides. I'm honored to be invited to this webinar talking about our ACE mass platform at WuXi. I'm going to talk about the principle of ACE mass, our workflow at WuXi, the power of using ACE mass, introducing an in-house case study, as well as a published paper of complementary using of ACE mass and other lead generation techniques. Affinity Selection Mass Spectrometry, which in short, ACE mass, is a high-throughput screening technology for drug discovery. It could rapidly screen a large collection of chemical compounds in order to identify ligands for specific biomolecular targets. ACE mass definitely gain more attention these days. Unlike other technology, the functional effects of this specific biomolecular target is not important in ACE mass library screening. In this case, novel ligand could be identified regardless of its binding site. So, how does it work? The small molecules and the target are first incubated together in solution, then separation is conducted and only leaving us the target with its binders. On top of that, dissociation step is carried out, and the binders will be analyzed by LC-mass. At WuXi, a hit identification of 20K library could be finished within 6 weeks. After receiving our biomolecular targets, assay development will be first carried out, including protein QC for testing the purity and activity. If positive control compounds are provided, tests will be run to validate as well. Incubation condition will also be optimized before the library screening. In the screening process, as many as 400 small molecules could be pulled together and incubate with the target. After the incubation, binders and non-binders will be separated using commercialized 96-well stack plate. Then the collecting plates will be transferred to our analysis group to identify the binders. ThermoQ Executive HF is used for this job. It is a high-resolution, high-sensitivity, and high-accuracy instrument and has done many brilliant work at WuXi. In the whole screening section, 2 rounds of the screening will be run. After the primary screening, the binders will be picked out and run for the second time, only in smaller pool. In order to lower the false positive rate and identify the true binders, 2 rounds of the screening done. The small molecule candidate can be then cherry-picked for binding assays or other functional assays, for example, KD test or IC50 test. So, why choosing ACE mass? ACE mass requires little for assay development. You don't have to know the binding sites. You don't even have to have a true binder as a positive control. And as I mentioned at the very beginning, there's no bias in target small molecule binding mechanisms. As we all know, a target's biological function could be elusive. However, it won't affect our ACE mass screening. And since it's in solution, all binding pockets of the target is opened up for small molecule mixture. It has more potential to identify a novel binding site. ACE mass is also scalable. In the case of WuXi ACE mass platform, as many as 400 small molecules could be pulled together and tested at the same time. ACE mass has broad applications. It works for any soluble targets and any library mixtures. Here's an in-house case study for you to get a clearer picture. In this case study, 300K library was screened for 2 rounds. The hit rate of the primary round is around 1%, and around 560 compounds were confirmed in the secondary screening. In the follow-up functional assay, all 560 hit compounds were tested in the radioactive assay, 22 compounds shows over 80% inhibition, and 20 compounds have the IC50 value lower than existing reference compounds. So pretty amazing results. And ACE-Mass could be coupled with other lead generation techniques. The paper shown in this slide published last year. Structure-based drug design and ACE-Mass were used to identify the new small molecule binders to the protein PCSK9. Compound 1 is one of the ACE-Mass hit compounds, and its R enantiomer could bind to the protein PCSK9 as the X-ray crystal structure shown. The ligand published in the paper itself did not display any functional activity, but with the targeted protein, the protein degradation will be utilized. ACE-Mass is a powerful technique in drug discovery, and I hope you enjoyed my talk. Thank you for listening.
Thank you, Penny, very much for this deep insight into ACMS technology. Now let us focus on the DNA encoded libraries. This technology is often seen as disruptive innovation. Why? DNA encoded libraries let researchers screen billions of compounds in a single experiment that is performed in a small Eppendorf tube, thanks to a DNA tag that encodes how each component in the library was made. Although the technology was invented about 30 years ago by Richard Lerner and Sidney Brenner, it became a mainstay in drug discovery only within less than years. So the enormous amount of compounds which can be screened at the same time have to be carefully designed. So how the library design and synthesis takes place. The most popular method for creating a DNA encoded library is called split and pull approach. It involves assembling DNA and a small organic building block. These components are split into wells, and another building block is added along with a second piece of identifying DNA. In this manner, it is encoded in the DNA tag how each component in the library was made. At WuXi AppTec, our building block collections include over 35,000 commercially available building blocks, which are combined with 6,000 scaffolds that were self-designed by WuXi AppTec's chemists. Thanks to this huge collection of building blocks and enormous chemistry power behind, we have created over 280 libraries in the size of over 65 billion compounds. This covers also a huge portion of the drug chemical space. Our libraries are pulled together in 4 different platforms. The Open platform includes open access libraries, which encode about 4 billion of compounds and can be purchased or they can be requested as a Dell Open kit and can be practically the screening performed by yourself. The Light kit contains 58 libraries encoding over 15 billion of commercially available compounds. And similarly as Dell Open, you can purchase this kit and perform the screening by yourself. Our largest platform, which includes over 150 libraries, is so-called Dell Pro. This is full service from the side of WuXi AppTec. This library contains 46 billion of compounds, including building blocks which are WuXi AppTec specific. I would like to point out at this spot that actually libraries are unique to the platform. That means that the libraries which you can get in the kit of Dell Open are not included in Dell Light or in Dell Pro. or the libraries which are included in the Dell Pro platform are not included in the kits. As a separate collection of the libraries, we have now also covalent library, which contains 50 plus libraries, just in the case if you are interested to find a covalent binder to your target. The workflow is actually very simple. Basically, this is a binding assay that looks like actually typical biochemical pull-down assay. So we are done with library production, and then we are good to go in the affinity selection. Very often we have to we use affinity matrix, let's say beads, which recognize specific tag on the protein, like his tag, biotinylated AVI tag, flag tag. And with the help of this tag, we actually immobilize the protein to the matrix. Then we add the libraries, incubate with the libraries, we wash off all non-binders, we elute the binders, and we repeat the procedure a few times. When we are happy with the number of enriched enrichment, we amplify this in the PCR step, prepare the samples for a next generation sequencing. Our informatics team decodes and translates the tags into the structure. And finally, the last step for the DNA campaign, DNA encoded library campaign, is of course the synthesis of DNA compounds and corresponding hit validation, either biochemical, biophysical, or cell-based. You can imagine that practically with this billion of compounds which are mixed in each, let's say, collection of libraries, we really need significant computational power to translate all the tags into the structures and analyze everything very closely. Basically, at the end of every screening, we get the information, which libraries were enriched under which conditions, how many copies of each compound we were able to catch, immobilize, practically catch with our protein. And based on all this information, very detailed statistical analysis performed, which actually results in a chemotype analysis, clustering analysis, and of course, the most important, we analyze all the hits based on their physicochemical properties and recommend the best candidates for the synthesis. As I mentioned at the beginning of this webinar, very often it's important to combine different lead generation techniques to get the best out of each technique. Nowadays, of course, is the computational power got actually very cheap. We have very powerful supercomputers, and machine learning is actually blossoming. Combination of machine learning and DNA encode libraries actually represents a new paradigm for hit finding. So basically, as described in publications cited here and shown here, based on the results of DNA encoded libraries, one can teach machine to recognize binders from non-binders, and based on all the chemical properties and how the compounds look like, we can actually use this machine learning to identify compounds from large libraries of commercially or easily synthesizable compounds. So as shown here, they actually, in this way, they selected about 2,000 compounds which were immediately available. You could purchase them from any of the catalogs. And from those 2,000 compounds, they had over 500 confirmed hits. And as you can see here, over 1/3 of hits had a KD of practically below 30 micromolar. This was one example. Next example is actually an interplay of lead generation technique. For example, as in this first-in-class RIP1 kinase inhibitor clinical candidate. Initially, at GSK, they decided to perform HTS. They found an excellent tool compound, but the process was kind of stuck there. So they decided actually to use a power of DNA encoder technology and search for the hits for this RIP1 kinase. They found a hit with high potency and selectivity, and also it has other proper characteristics, and also like for example, potential to achieve desired oral exposure in rodents. So how come that they were able practically to pick up this good candidate? Yeah, basically on the chemistry which they have observed on the hits coming out of HTS, they practically knew which hits are not so interesting, or maybe let's say a dead end within the DEL hits. So they actually were able to make an educated decision on which hits to work and to continue. And as you can see here, only with the minor chemical change, this hit from DNA encoded library screen was appropriate clinical candidate. So next, practically now we are going from billions back to thousands. And as mentioned, as mentioned previously, ACMS and DNA encoded libraries mostly deliver a drug-like hits which bind into nicely formed crevices or binding pockets on the protein. Some proteins lack clearly defined binding pockets, or maybe we want to target something on the surface of the protein where practically no obvious binding pocket is formed. In this case, a use of tiny fragments might be an approach of choice. So Saskia, please tell us a little bit more about fragment-based drug discovery and what are the advantages of fragment screening.
Thank you, Nusha. So what are the advantages? Why do we offer fragment screening? One reason is that it is complementary to classical HTS and DEL screening. In fragment screening, we work with rather small molecules with a size of 200 to 300 Dalton. Due to their size, they can bind to small pockets available on protein surfaces and thus provide a good starting point for inhibiting protein-protein interactions. One big advantage is also that fragments are quite simple chemical starting points. This allows for more flexibility during hit-to-lead optimization. The fragments can be grown, linked, or merged. And this allows a very efficient way of sampling chemical space as well. The chemical simplicity also allows efficient optimization of pharmacokinetics in parallel with potency, as the fragment hit grows into a clinical candidate. And finally, hit rates from fragment screening are excellent indicators for the druggability of a protein target. So our typical workflow looks as following. We perform the primary fragment screen at one concentration by one technique. So this can be MST or TRIC or SPR. TRIC is the phenomenon predominant on the Dianthus, the second generation instrument of MST. Then we perform an affinity ranking with the same technique. So again, MST or TRIC or SPR. This is followed by orthogonal hit validation using a different technique, for example, nano-DSF, but it can also be ITC or NMR. And with the hits, we attempt structure generation to allow structure-based fragment-to-lead development. If this is not possible, we can use activity-led fragment-to-lead development. So now I want to give you a more detailed overview of our screening workflow. First, we produce the protein and run a QC by SDS page, analytical sec, and mass spec. Then we use nano-DSF to optimize the assay conditions. We screen for an optimal assay buffer, detergent, and DMSO concentration. We also confirm protein activity by analyzing tool compound effects on the protein thermal stability. Based on this information, we perform an extended assay setup. We investigate which technique, MST, Dianthus, or SPR, is most suitable for the screening. Once a technique is identified, we run a single point screening, typically at 500 micromolar, followed by an affinity ranking of the identified hits. So this whole process typically takes 3 to 5 weeks. Afterwards, the identified hits are validated by an orthogonal biophysical or biochemical assay. We also recommend to set up a suitable crystallization system in parallel because we are all aware that structure-guided drug discovery is the most efficient way forward. And further down the road, we can also do SAR by catalog or in silico approaches. So on the following 2 slides, I will show you some real-life data of a case study that we performed with the stimulator of immune response genes, in short, STING. The construct was designed with a HisTag and an AVITag, such that fragment binding could easily be analyzed by MST and SPR. For MST, we established non-covalent labeling of the HisTag with Retris NTA dye. The assay was validated by demonstrating reproducible binding of 2,3-CGAMP, the natural substrate of STING, and we found an affinity of 10 nanomolar, well in line with published data. We ran then a primary screen at 1 concentration, followed by a KD affinity ranking. The best fragment hit exhibited binding with KD values in the 2 to 3-digit micromolar range. For orthogonal hit validation, we made use of other assays that we have established in-house. On one hand, by SPR, immobilizing STING to streptavidin chip via the biotinulated AVITag. On the other hand, nano-DSF. By that, we were able to confirm 66 binders. We also decided to validate a subset of these binders by STD-NMR, and the data was really encouraging. 9 out of 11 selected hits were confirmed by NMR. And with this, I'd like to thank you for your attention, and I hand over back to Lucia.
Thank you, Saskia, for these interesting examples. The first FDA-approved drug, verumafirib, PLX4720, is an inhibitor of BRAF enzyme developed by Plexicon and Genentech for the treatment of late-stage melanoma, demonstrated the power of fragment drug discovery for the target, where achieving the selectivity can be a real challenge. And as you can see here, practically the clinical candidate was developed after switching the selectivity from PIM1 over FGFR1 and finally to BRAF mutant V600E. As you can see here also on this slide, the FBDD process is tightly defined by a close interaction between biophysics, structural biology, and medicinal chemistry and computational chemistry as the fragment grows toward the potential clinical candidates. Thanks to enormously increased computational power, computational chemistry entered a new era and offers a variety of approaches which lead to the innovation in the drug discovery at a faster pace. Link. In which cases would you recommend virtual approaches, and what is today's state of the art?
Virtual screening has been widely applied in early-stage drug discovery as an alternative or complementary approach to high-throughput screening assay. Virtual screening is a knowledge-driven compound database searching approach which attempts to find novel compounds and chemotypes with the required biological activities. If the three-dimensional structure of a target is not available, ligand-based virtual screening can be applied as a compound selection filters for identification of biologically active compounds. When the 3D structure of a target protein is known, both high throughput docking and receptor-based pharmacophore virtual screening approach can be applied for identifying new structure candidates. Virtual screening can be carried out against both real and virtual library. There are docking-based, pharmacophore-based, shape-based, fingerprint-based, and machine learning-based methods available for virtual screening. Docking-based and pharmacophore-based methods requires that receptor crystal structure is known. It is computationally expensive and can process up to millions of compounds. Shape-based and fingerprint-based methods does not require receptor structure to be known. It is not computationally expensive to run, and it can process billions of molecules in short time. There are various virtual screening workflow available. This slide shows one of the virtual screening workflow provided by Schrödinger's glide. Once the receptor docking model is built, you can use Schrödinger's virtual screening protocol to carry out virtual screening against any compound database. In the example on the left side of the slide, a collection of 2 million compounds has been run through the glide virtual screening workflow via HTVS, SP, and XP docking steps to filter down from initial 2 million compounds to 20,000 compounds. The right side shows a typical pose distribution over XP docking score. Furthermore, more accurate MMGBSA can be used to rescore the top XP docking score compounds shown in the orange box from the results of the previous virtual screening. The success of virtual screening depends on many factors. One of them is how large the chemical space it can access. Virtual libraries are able to store more compounds than can be held in physical format. Make-on-demand libraries are typically made by one-step or one-part reactions. Because of the availability of tens of thousands of building blocks and hundreds of reactions, make-on-demand libraries are diverse, growing exponentially, reaching the range of billions. It becomes a perfect library for virtual screening. On the left side, the graph shows make-on-demand library only has less than 10 million compounds around 2015. But the number of the compounds in make-on-demand library are projected to over billions by 2020. The right side graph shows the distribution of compounds among 10 million scaffolds in the make-on-demand library, which indicates make-on-demand library is diverse. One of the examples of make-on-demand library is WuXi AppTec's Galaxy library. WuXi AppTec offers one of our own make-on-demand library, which is called Galaxy. Galaxy has over billions of accessible small molecules, which can be accessed through virtual high-throughput screening or with biosolver IT Infinity. Galaxy is built with unique scaffold templates and building blocks. Its compound usually can be delivered in 4 to 8 weeks. The library has the right diversity and with the right physiochemical property for drug design applications. As virtual library continue to grow, conventional docking-based virtual screen can fail to capture an expansive breadth of chemical space. In contrast, machine learning approaches afford the opportunity to rapidly and inexpensively explore vast chemical space in silico. Virtual high-throughput screening as an established methodology is being transformed by the fast development and application of machine learning algorithm. With machine learning technology, virtual screening is no longer limited to conventional molecular docking for targets with good structure information. Also, machine learning expands the scale of virtual screening in a revolutionary way. Key features of machine learning empowered virtual high-throughput screening platform include, first, the platform can vastly expand chemical space that is limited by real compound collection before, via its ability to process large chemical space from millions to billions, and its ability to run in silico screening with on-the-fly enumeration and make-on-demand virtual library. Also, the platform can reduce real laboratory experiments and transform drug discovery in a more efficient and economic way, such as provide rational starting points for integrated service, or evaluate target binding with a high successful rate of activity. This cell paper from MIT group provided an example of machine learning empowered virtual screening. The researcher first trained their neural network model using a collection of 2,335 diverse molecules for those that inhibited the growth of E. coli using a directed messenger passing approach with building a molecular representation based on a specific property. Next, they applied the model to multiple chemical libraries with more than 100 million molecules to identify potential lead compounds with activities against E. coli. After ranking the candidates according to the model's predicted score, a list of promising candidates was selected for bioassay. The compounds which they identified demonstrated efficacy in mice infection model. Also, their deep neural network model has shown to identify diverse drug-like and easily synthesizable molecules that are different from the original screening libraries. In 2019, the researchers at UCSF for the first time has demonstrated the docking of ultra-large libraries for chemotype discovery. The author built a virtual library of 170 million make-on-demand compounds from 130 well-characterized reactions. The resulting library is diverse, representing over 10 million scaffolds. The library was docked against AMPC beta-lactamase and D4 dopamine receptor respectively. Against the AMPC, they docked the make-on-demand lead-like library, which composed of 99 million molecules. These molecules were docked and 44 compounds were synthesized and tested. This revealed an unprecedented phenolate inhibitor of AMPC, which was optimized to 77 nanomolar, which is the most potent non-covalent AMPC inhibitor known. Against D4, over 138 million library molecules were docked against the structure of D4 dopamine receptor. 549 compounds has been tested at 10 micromolar. 30 new chemotype of sub-micromolar activity were found. A subtype selective D4 agonist with 180 picomolar has been identified from this exercise. Crystal structure of 3 of the new ligand family and the ligand of the 77 nanomolar were determined to a resolution ranging from 1.5 to 1.91 angstrom. Unanimously, electrodensity confirmed their fidelity to the docking prediction, with the IMSD varying from 0.98 to 1.2 angstrom. High fidelity between docking and X-ray crystal structure was observed. Docking of ultra-large library can be done and identify new chemotype, as demonstrated by the previous example. However, the approach is time-consuming and requires large computational resources. The researchers from University of British Columbia has introduced the deep docking for rapid screening of ultra-large library, and it does yield potent novel compounds. Deep docking is a novel deep learning platform which is suitable for docking billions of molecule structures in a rapid yet accurate fashion. The deep docking approach utilizes QSAR deep models trained on actual docking score of small subset of a chemical library to approximate the docking outcome for yet unprocessed entries, and therefore to remove unfavorable molecules in an iterative manner. They also demonstrated that the use of deep docking methodologies allows rapid and accurate calculation of docking score for 1.36 billion molecules from the zinc-15 library against the 12 target proteins, and demonstrates up to 100-fold data reduction and a 6,000-fold enrichment of high-scoring molecules. The deep docking approach circumvents computational limits of large-scale docking campaign and makes billion-entry molecular database accessible even with limited computational resource.
Thank you, Ling, for the deep insight into the world of computationally assisted drug discovery. I think that we are all impatiently looking into the future and waiting on the clinical candidates which will arise from AI and machine learning. Now, at the end, I would like to quickly summarize what we have learned today. We know, I hope, that practically we were able to demonstrate you that different target types require different hit finding technologies to find the best chemical matter. And different technologies will uncover different chemotypes. Still, we should not focus only on the quality of the libraries. We have to pay the great attention to the protein. Early parallelized exploration of various target protein variants can gain several months in overall project timeline. And most importantly, and I really hope that we were able to show you, based on success stories, the synergy between various lead generation techniques efficiently drives innovation. Overall, in less 20 years, evolution of lead generation toolbox led to enhanced success of drug discovery in the hit-to-lead process. Thank you very much to your attention, and now we will answer your questions.
Thank you so much to Nuska, Penny, Saskia, and Ling for that excellent presentation. And now we're going to open it up for questions. This question is for Nuska. How can we know that in the release process from the protein, the target molecule is not modified?
So let's say our DNA encoded library collection is, we separate practically the covalent libraries from non-covalent libraries. So practically when we use, if I may say, classical libraries, we know that target molecule will not get modified because our, so to say, the ligands are simply not chemically reactive. But of course, if you're searching for the ligand which covalently modify your target, then of course we can do using our covalent libraries. And in that case, of course, the target molecule will be modified, And in that case, we have, of course, to adjust experiments accordingly.
Okay, thank you very much. This question is for Penny. Hopefully you can hear us okay. Since compounds are identified by their molecular weights and many different compounds may have the same molecular weight, is it possible that compounds with the same molecular weight may be missed? And how is this problem handled?
Thank you. Thank you for the question. And for this question, our compounds that have the same molecular weight have different structures. So their RT, which is retention time, are different on our output column. And the software can deal with that under this circumstances. But if the compounds were isomer, the problem you worry does occur. So when we prepared our library, we always try to avoid putting the isomer in the same well. And over 90% of the compounds in our library can be detected out. And thank you.
Thank you very much. This question is for Saskia. What is MST?
Yes, thank you for that question. So MST stands for microscale thermophoresis. So it's a technique developed by Nanotemper Technologies. It's based on an equilibrium method that you can use to determine affinities of small molecules to protein targets, or also protein-protein interactions, or any biomolecules that you can think of. So it is based on a change of the mobility of your target in a temperature gradient. So what you do is you incubate your target with different concentrations of a ligand, and then you check how does the presence of the ligand influence this thermophoresis, the mobility in the temperature gradient, and from that you can extract a KD value for the dissociation constant. And this can work with labeled targets or with unlabeled, and the Dianthus instrument that I mentioned, or the trick, is the further development of that, because the MST uses a capillary format and the Dianthus instrument uses a plate format. And both of them can be used for screening of large molecules, large number of molecules, such as fragment screening.
Okay, thank you very much. This question is for Penny. How do you pick your hits?
Okay, so for HPF techniques like ACE-Mass, we often use software to identify our hit compounds. We set up our parameters and process our data. And thank you for the question.
Okay. This question is for Nuska. Is DEL applicable to RNA or DNA targets?
Yes, indeed, DNA encoded library technology is amenable also for RNA and DNA targets. And you can imagine these are difficult targets, so we really have to carefully plan the experiment and all the controls. But in any way, we can successfully find ligands for RNA and DNA targets.
Okay, great, thank you. This is also another question for you, Nuska. Can you talk about the relationship between DEL hits and clinical candidates? How much optimization is typically required?
Thank you for actually this tricky question. It's always case dependent. Yeah, so seldom you will practically get the clinical candidate directly from the, let's say, DNA encoded library screening. Yeah. On the other hand, if you are lucky, you can just with a few modifications, you can be already there where you want to be. Yeah. So unfortunately, the answer is it's a case dependent.
Okay. We have time for about 2 more questions, and I believe they are for Saskia. How to follow up fragments if no structural information is available?
Yes, so we can do SAR by catalog. So we check for available compounds and do an SAR on the compounds.
Okay, and this is our last question. Oh, I'm sorry. Go ahead, please, if you have more to elaborate on.
Or, of course, in silico methods. I just wanted to add a little.
Okay, thank you. Here's an additional question. How long does it typically take to develop a lead molecule with NM affinities after a fragment screening?
With nanomolar affinities, I guess that's supposed to mean. So because
yes,
the fragments that we the fragments that we get as hits, they have 2 to 3 digit micromolar affinities, but of course from that you want to develop a nanomolar affinity binder. That will take quite a lot of work, and what we typically expect is that it takes more than 1 year to get to this good affinity range. So this is something to keep in mind before going into a fragment screen.
Okay, thank you very much. And that's about all the time that we have today. Thank you so much to Dr. Nuska Schemmer, Dr. Penny Jha, Dr. Saskia Villinger, and Dr. Ling Xu for your presentations. And thank you, participants, for being a great audience today. Be sure to check CNEN or CNEN Online for information on the next edition of CNEN webinars. Thank you to On24 for the technology and production services, and a big thank you to WuXi AppTec for the sponsorship that made this interactive webcast possible. For CNEN webinars, I'm Melissa O'Meara. Goodbye.